Okay. I've also turned on the recording. Yes, I've turned on the recording. So before we start the class, there is an announcement for your upcoming minor exam. So you must have seen your timetable, your exam schedule, and your exam for this slot is on Tuesday, the coming Tuesday, and we will have the exam at 2 p.m. Is that time suitable to everybody? Your exam has to be conducted on the coming Tuesday and has to be completed by 3 p.m. So I suggest that we start the exam at 2 p.m. I will upload the question paper on Moodle. I hope you're all aware of the platform Moodle. So I will upload the question paper on Moodle, and I will also send a copy of the question paper by email. You have to write down the answers. Either you write it down or you type it, whatever is the method that you choose. Either you write it down or you type it. Type your answers, write down your answers. If you're writing down the answers on a piece of paper, you take a picture of that with your phone, or if you have a scanner at home, scan it and send it to me by email. If you're typing the answers directly on your computer, send it to me, whatever it is, whatever the method of writing down your answers, send it to me by email. And there will be a time stipulated that there is going to be this much amount of time for the exam. So I will give you an additional 5 minutes so that I can receive all the emails. Once I've received the email, I will send you back a reply saying received. If you do not see that reply, you must become aware that I have not received your answer scripts and you must get in touch with me immediately. If you do not see the reply mail saying received, okay? And the exam will be an open book exam. So you will be allowed to look through your class notes or even internet to find the answers. All right, any other questions related to exam if you have?
Exam is set for 2 hours.
Huh?
Exam is set for 2 hours.
No, it's not set for 2 hours. It's not set for 2 hours. It would be somewhere around 1 hour, maybe even less, maybe 40 minutes, 40, 45 minutes, something like that. But the exam has to be completed by 3 p.m. That's the stipulated time on the exam calendar that is given by IIT. So it has to be completed by 3 p.m. of Tuesday. And nobody has to come to the classrooms to give your exams. Even if you have onboarded for this particular course, you can give your exams from your hostel. Even if you are staying in hostels, you can give it from your hostel. No need to come to the classroom for the exams. All right. I hope that is okay. So let us start the class. All right, today what we are going to do is give ourselves a little bit of recap of kinesins and what we have studied about kinesins, and then to look at one interesting aspect of kinesin. So, kinesins, if you remember, we were looking at the reasons, or people were looking at the reasons why or how basically you would be able to transport materials across long, very long distances of a cell. And when they are talking about long distances of a cell, they are not talking about cells of micrometer order distances. Of course, that's also a pretty considerable distance, uh, considering the size of a protein would be somewhere around in nanometers. But let us consider the case of a neuron, and a neuron whose axons are, whose cell body is somewhere in the spinal cord here, and but whose axons run the length of your leg and reach the tips of your foot. So for an axon of that length, how is it that materials are transported from the cell body to the tips of the dendrites present at the foot? So that was the question, and they were studying this using different materials. And as we had seen the last time, there was a video where you could see that if you add beads, carboxylated beads into a cell by some means, say you micro-inject it into a cell, you would see that the beads would start moving. And they seem to be moving along tracts of microtubules, as can be seen here. This is on a slide. So you see tracts of microtubules and beads are moving on those tracks with time given here in seconds. And here you can see similar movement of organelles. There is a bulge here and this is an organelle that is moving on a microtubule track. So there is something that is guiding these beads or these organelles on microtubule tracks. So microtubules are used as guiding tracks, but there's something that is moving it along these microtubules. And therefore there must be a motor doing it. So initially people had thought that it would be myosin. Turned out that it is not myosin. Then there ensued a race to identify the exact motor protein that is causing this. And in the meantime, before even they identified the motor protein, they realized that the motor protein, whichever protein was doing this, had a dependency on ATP. So, for example, if they added a non-hydrolyzable ATP analog, which we have named it here, if they add this analog, then the movement of these beads on the glass slide. So this is an experimental design. What's the experimental design? The experimental design is you take a glass slide and you add microtubules to the glass slide so that you will see single tubulins, cylinders lying on the glass slide. Now you add the carboxylated bead, and it's not enough to just add the carboxylated bead. You need to add something else. Can somebody remember what else did we add to this system so that the beads moved? Because if you just take purified beads and you take the tubulin, the beads will not move. What is it that was added so that the beads started moving? No? Okay. So what was the initial system where this entire thing was identified? It was identified in axons, right? Squid axons. So in squid axons, if you add these beads, you will see that inside the squid axons, the beads will start moving. So if you now want to replicate the same thing, what you can do is you break up that axon, squid axon, you break up those cells and you take the cell-free extract. So that should have all the proteins, sort of motors, whichever motor is required to move that bead along the microtubule tract, that should be present in the cell-free extract. So if you take the cell-free extract, combine it with the beads, add it to this add it to this glass slide that has the microtubules on top of it, then you should see the beads would start moving. So this is a very simple assay to see the movement of the bead with the help of these motors. They didn't know what the motor was, they knew that something like this can be done. But it was not so simple. The first thing they found was that the beads didn't like attaching to microtubules. They didn't. So if they added this ATP, non-hydrolyzable ATP analog, that's when the beads had sort of a very stable attachment to the microtubules, but the beads didn't move. So there was some dependency on ATP, but ATP hydrolysis was required for the beads to move. So adding the non-hydrolyzable analog of ATP ensured that the beads stuck very well, very nicely to this microtubule, but didn't move, didn't translocate. For a translocation, ATP hydrolysis was important. So if you could inhibit ATP hydrolysis using very high concentrations of a compound, which is given here, vanadate, if you use this, again you will be able to stall the beads, that is, the beads will no longer move, which indicated again the same thing. You need ATP hydrolysis for the movement of these beads. Then the question comes up, what is it that is there in the extracts? This is the extract. So if you just take a cell, any cell, you take a cell, you lyse the cells, and you take out all the proteins from a cell. You run those proteins on an SDS page, you will see multiple bands of proteins. And each of these bands, in fact, can be more than one protein because there can be more than one protein of the same molecular weight. So each of these bands need not be one protein. In fact, they are not. Mostly they are composed of all the proteins that fall into that narrow range of molecular weight. So they will form this one band. You have multiple bands as such in in an axonal extract or in a squid extract. So now the question was to identify which of these proteins is the actual motor here. And they took the help of the bead assay, because if it can bind to the bead, right, if it can bind to the bead, then there can be a way to purify it from those beads. So they used microtubules here alone, squid extract alone, mix these two, okay, mix these two so that these microtubules can be now bound by the motor protein. Now, what is the next information they had? The next information they had was that if they add this non-hydrolyzable analog, the beads bind very tight to the microtubules. They are not released. That means the motor proteins that are attached to the beads, they are bound very tight to the microtubules. So if I want to identify the motor proteins, I have taken this extract, I have taken microtubules. Now if I add the non-hydrolyzable ATP analog, then my motor proteins should be bound very tight to the microtubules. It will just be a matter of purifying those microtubules out of solution or extracting them, because now when you extract them, the bound motor proteins will be extracted out too. So they did that, and they found that there was a faint band here. That this process they could repeat, enrich it multiple times to get this 110 kilodalton band. And this 110 kilodalton band, you could increase the affinity of the motor protein for microtubules by increasing the amount of ATP. So successively, if you keep increasing the amount of ATP, you get more of this motor protein. So this was the really first experiment that showed how you could identify motor proteins that are involved in long-range axonal transport, and they called them kinesins, like kinetics, kinesin movement. So, unlike myosins, kinesins turned out to be a huge family. So it's called the kinesin family, superfamily, and we found that there would be about 45 genes. Now, this is work that I would give you after the minors, where I would ask you to I'm sure all of you know how to use NCBI and obtain gene sequences. So if you just take the mammalian or human as the organism, and you now try to find out all the kinesin genes that are present in humans, and if you then try to identify what is if you can group them into families, you would see that they would really fall into certain patterns because of homology in sequences. So I'll give this to you after the minor, but for the moment, let's look at this slide. You see that there are a number of these kinesin genes that are known, and each of these genes can undergo alternate splicing, so you can have multiple kinesin proteins. So 45 genes, but more than 45 proteins. And what is common about all of them is that they have a motor domain and they have a coil domain. So this coil domain is the domain that helps them form dimers or multimers. So if there are two dimers that are walking, then the dimerization has to happen with the help of this coil domain. And coil domains are also used in experimental system to artificially induce dimerization. Suppose you want to study a protein on a cell membrane, and you know that the protein is active when it is dimeric. So if you would want to induce artificially a dimerization, you can simply add coil domains to the transmembrane region of the protein. You will see that they will dimerize. So Coil domains in general are there for this dimerization effect. They are used in nature. They can also be used by you when you would like to design experiments to induce artificial dimerization. Add coil domains, you will see that they will dimerize. What do we see? We see that there is a motor domain, there's a coil domain, and the motor domain, of course, by now you know that it's the domain that is required for the motor activity, that is for its motion on the microtubules, whereas coil domains is for dimerization, right? Okay, we also found that unlike the general idea that dinings are this is a general idea and I'm not saying that it's wrong. Okay, don't take it that way. It's not that it's wrong, but the general idea is if you have a big cell and you need to transport proteins, you need to transport mRNA or you need to transport organelles from inside the cell to the periphery, that is from close to the nucleus to the cell membrane, then those molecules or those organelles will be moving from the minus end of a microtubule to the plus end, that is the growing end of a microtubule, right? And this plus end movement is done by kinesins. Whereas if you are looking at things which are outside the cell and the cell needs to ingest them, so there is endocytosis and those materials need to be brought inside the cell because they have to be processed, then those would be moving from the plus end of a microtubule to the minus end of a microtubule, and that would be done by another class of motor proteins called dyneins, which is true, which is a general idea. But we have seen last time that your kinesin motors can walk towards the plus end, can walk towards the minus end, and can also depolymerize microtubules. Now, depolymerization of microtubules is something we already saw in our classes when we were dealing with microtubules, that microtubule protofilaments tend to bend. That's the dynamic instability, right? So they would grow, they would grow, they would grow. At some point, you'll see that the protofilaments bend, and the bending causes dynamic instability. The entire microtubule sort of depolymerizes, and then the polymerization starts again. So the first thing here is that any protein that can induce the bending would cause an increase in dynamic instability, would cause the protofilaments to sort of break apart. And we found a kinesin motor could do this. So we saw how a kinesin motor, it can bind to the protofilaments and cause bending of the protofilament, like a curvature in the protofilament. So that's one part that the kinesins can do. Another part that we talked about is that kinesins can walk both on towards the plus end of a microtubule end. So that's the conventional kinesin, but there are kinesins that can walk towards the minus end too. And today we'll have a brief look at this property. The other thing that we learned last time was that kinesins can interact with various kinds of cargoes, right? So how does it pick out its specific cargo? How does a kinesin know which cargo to transport? So we do not know the entire answer. It's a big puzzle because of course, there are so many molecules, so many different kinds of organelles, mRNAs, etc., to be transported. What we do know is that it could be dependent on the heavy chain of kinesin molecule itself. So the one end of the kinesin is walking on the microtubule. The other end could be itself interacting with the cargo, or you could have some light chains present at that region, and those light chains could be interacting with the cargo. You could have some adapter molecules. Those adapter molecules could be interacting with cargo. So there are different ways by which these kinesin motors can interact and can carry the cargo. And we also looked at the structure of the motor domain the last time, and we saw that in the way the kinesin motor works is quite different from the way the myosins work. In the sense that in myosin, when myosin motor head is bound by ATP, that's the point the myosin head can no longer interact with actin, right? So it detaches from actin. But The myosin head has an intrinsic ATPase activity, so ATP is hydrolyzed to ADP and inorganic phosphate, and that creates a loose attachment to actin, and so it then binds to actin. Here, on the other hand, what we find is that binding to ATP, and not the hydrolysis, just the binding to ATP itself is enough to cause a conformational change, in the sense that this region, which is called the neck region and the neck linker region, that forms right before the coil domain starts. So somewhere here, this region, the neck and the neck linker region, is kind of perpendicular to the motor. But the moment ATP binds, there is a groove that opens up slightly, allowing this neck and neck linker to bury itself inside the motor domain. And this causes the characteristic walk of the kinesin. So you have this head, the motor head that binds to microtubule. ATP is bound, and now that causes the neck linker to attach itself into the groove that flips the whole molecule forward. Another movement, another linker burying itself, flipping off the other foot. So it's almost a hand-on-hand movement. So when people were trying to understand how does the kinesin move, there were a lot of hypotheses. One was that, okay, if there are two such legs, then one way it can move is that, okay, it can move like a hand or over a hand over a hand movement, that's what it is called. Or it could be a worm-like movement. So if you've seen caterpillar worms, what they do, they move like this, right? They move like this, they inch forward slowly. So it could be that movement. So then they did experiments. Of course, you know what kind of experiments it could have been. It's of course the optical trapping experiment that would give you an idea of how much displacement it is causing with time. So you'll be able to understand whether this is a hand-on-hand movement or it is a worm-like movement. And they found that this is a hand-on-hand movement. They combined that with cryo-electron microscopy, with capturing a lot of these images, then reconstructing the way these kinesin motors move to finally get this picture. Okay, and we also found that because there is this kind of a movement, which means that it sort of utilizes the periodicity that is present on a microtubule. So why is the microtubule periodic? Because you have these alpha beta dimers which are polymerizing end to end to give you a microtubule. So there is a periodicity, and the kinesin motors follow that periodicity. And so each step here is at an interval of 1 dimer. So that translates to about 8 nanometers of step size. Okay. Now, today the question that I would like you all to ask yourselves is that we have seen so far that kinesins can be dimeric, but we've also learned last time that some of the kinesins can be monomeric. So it is easy to understand or visualize a kinesin which is dimeric, which has 2 foot and is using one hand flipping over the other. Then the ATP binds, this hand flips over, and it walks on the microtubule, right? Very processive movement, can walk considerable distances without actually falling apart from the microtubule. But what if it is a single chain of kinesin? It's not dimeric. What happens to kinesins which are monomeric? How do they walk? Because then you cannot envision that you will have 1 foot and then there will be another foot that flips over. There's just 1 foot. There's just one chain. How do those motors move? And what do we know about those motors so far? It's, as I said, it's easier to understand about a double-headed motor. What about a single-headed motor? So any idea, anything that any one of you might know? No? Okay. More time, let's go straight. Okay, so here we are taking an example of a kinesin motor, which is known to work as a monomeric protein. Because our question is, if it is a monomeric protein, then how does this monomeric protein behave? Because it doesn't have the other hand to flip over, or it doesn't have. See, the whole point is when we walk, how do we walk? We do not have at any given time point, we will not have both our foot in air, right? एक पैर जमीन पे होता है, दूसरा पैर हवा में, फिर वो पैर नीचे आता है तो दूसरा पैर हवा में जाता है। ऐसा कभी नहीं होता कि दोनों पैर एक साथ हवा में, हम वैसे नहीं चलते। तो अगर हम वैसे नहीं चलते, क्यों नहीं चलते? क्योंकि स्टेबिलिटी नहीं रहेगी। हमें तो एक्रोबैटिक होना पड़ेगा अगर वैसा करना है तो अगर हम यही चीज काइनेसिन के लिए सोचे कि दो पैर नहीं है, सिर्फ एक पैर है, तो वो चल कैसे रहा है और माइक्रोट्यूब्यूल से वो गिरने क्यों नहीं जाता, और क्या वो गिरता है? अगर वो गिरता है तो क्या वो गिरता है? अगर नहीं गिरता तो वो क्यों नहीं गिरता? This is the question that we are asking. Very simple question. So we take this. So this is an old paper. You can see 1999 science paper that dealt with this, and there were a couple of papers back to back during that time that dealt with the same question of how do these monomeric kinesins actually work. And here is one family of kinesins which is known to act as a monomeric kinesin. That is, it just has one foot to hop on, the microtubule. Now, because this is a big molecule to deal with, what they did in this experiment, in this particular paper, is that they truncated it. So truncation किस तरह से? ये है आपका काइनेसिन, राइट? ये तो डबल हेडेड हो गया। आप सिंगल हेड सोचिए, single head and single chain. So what is the minimum unit that is required for this kinesin to bind to a microtubule? Just the head and a little bit of neck linker is enough. Doesn't need this, because ये तो dimerization के लिए चाहिए या एंड में शायद कार्गो बाइंडिंग के लिए चाहिए। अगर मैं कार्गो बाइंडिंग डायमराइजेशन हटा दूं तो भी ये माइक्रोट्यूब्यूल से बाइंड करेगा। So this is what they did. This C351 is the name they have given. Is what? It's just that it contains the catalytic core of the motor domain followed by a short linker. Enough for it to bind to a microtubule. Abhi kya hai? Now you take this K381. What it is something that has a coiled domain. So if you now add a coiled domain to this, you can make it dimerize. So you have two conditions. आपने दो कंडीशन क्रिएट कर लिया। एक तो है सिर्फ हेड डोमेन लिया, थोड़ा सा लिंकर, नेक लिंकर रीजन रखा ताकि वो माइक्रोट्यूब्यूल से बाइंड करे, किसी भी तरह डायमराइज ना करे। अगर आपने इसके ऊपर कॉइल्ड डोमेन डाल दिया तो वो डायमराइज हो जाएगा। सिंपल कंस्ट्रक्ट, वेरी सिंपल कंस्ट्रक्ट। So you have created these two conditions where you have one which is absolutely monomeric, you have another which is dimeric, two conditions, and you have labeled these motor proteins with a red fluorescent dye and microtubules with a green fluorescent dye so that you're able to see it under a microscope। तभी उनको क्या दिखा है? उनको ये दिख रहा है, these are microtubules, so these are like three different snapshots. This is one snapshot, this is one snapshot, and this is one snapshot. The three different snapshots, यहाँ पे उन्हें क्या दिख रहा है? There is a microtubule which is in green, right? There's microtubules which is in green. And in red you have the motor protein. Which motor protein are they looking at? The motor protein C351, जो हमने पहले कहा है कि सिर्फ मोटर हेड है, थोड़ा सा लिंकर रीजन है, it cannot dimerize. And what did they see? If they add high concentrations of the motor, so high concentrations मतलब कितना concentration वो भी इन लोगों ने mention किया है। The moment they add high concentrations of the motor, you expect too many motors to bind to the microtubule. And if the motors are moving in the presence of ATP, then they would move towards one of the ends of the microtubule, because it could be a plus end directed kinesin, or it could be a minus end directed kinesin, whatever it is, it would move towards one end of the motor tubule, of the microtubule. And that is what they found. With time, all of these red color ends up staying at one end of the microtubule. So you see, this is a microtubule. Microtubule का thickness almost same है, लेकिन kinesin on this microtubule with time seems to end up at one end, almost like a lollipop. So this is another microtubule, it seems to go towards the end. This is another microtubule, it seems to go towards one end. So the first indication here is that even if you have a single microtubule head, it is enough for that microtubule to, it is enough for that kinesin to start walking on the microtubule and then accumulate at one end, because it has walked, all of these kinesins, single-headed kinesins have walked on the microtubule and have started accumulating at one end. अभी अगर हम इसको फॉलो करें एट लो कॉन्सेंट्रेशंस, ये हाई कॉन्सेंट्रेशन में था, सो यू हैड मल्टीपल kinesin units binding to microtubule. अगर हम लो कॉन्सेंट्रेशन में कर दें ताकि we are able to follow one single head that is walking on a microtubule, how would that look? How would that look? So they did the same experiment, and what you see here are snapshots of kinesin head walking over a microtubule, and each successive snapshot has an interval of 0.5 seconds. So it's almost like they have taken a long video, but video क्या होता है? Video होता है successive frames को जोड़ के video बनाया जाता है ना, तो successive frame को आपने 0.5 second पे एक frame लिया है. And then you combine that and you try to understand how is this single head moving on the microtubule. Sorry. So now let us look at this. You have the microtubule which is in green and you have kinesins in red. अभी ये arrow head, सिर्फ जो arrow head है इसको देखिए और ये जो arrow पे है इसको देखिए. So there are two different kinesin heads. Single-headed kinesin, not the double-headed kinesin. Single-headed kinesin, जिसका coil domain नहीं है, नहीं dimerize कर सकता at any consequence, it cannot dimerize. तो आप देखते हैं कि ये जो फ्रेम है यहाँ पे आपको दो दिख रहे हैं, दो ऐसे मोटर्स बाइंड कर चुके हैं माइक्रोट्यूब्यूल पे. लेकिन इसके पहले वाले फ्रेम में देखिए यहाँ पे एक माइक्रोट्यूब्यूल, एक मोटर यूनिट था, इससे भी पहले एक मोटर यूनिट था. So this is one motor unit that is moving. This is a second motor head that has bound and that is also moving. So अगर आप carefully notice करें तो between successive frames you can see that this motor head is moving in this direction. It's constantly moving in one single direction, है ना. आपका ये जो arrow head है ये move कर रहा है. But if you look at this, you see initially it has moved a little towards this side, then it moves back to this side. So ये भी move कर रहा है लेकिन ये पहले थोड़ा पीछे की, it's almost like ये पहले थोड़ा पीछे की तरफ गया है फिर ये सामने की तरफ गया है. तो इसका मतलब क्या है? इसका मतलब ये है, there are two things that are important from this experiment. One is that a single head of kinesin can sit on a microtubule, attach firmly to it and can move. And the movement is I would say quite processive. It's not like it's taking one step and falling off. And then you have the second kinesin that attaches to it and then you're actually tracking that kinesin. No, it's the same kinesin and that seems to walk considerable distance on the microtubule. It doesn't quite fall off. But what is it that is different about the single-headed kinesin? It sometimes sort of backtracks, sometimes sort of goes behind, then it comes forward. Which means कि इसके directionality में थोड़ा problem है, क्योंकि ये इसका जो directionality है वो कैसे determined होता है? It's all determined by the binding of the ATP, the locking of the neck and the neck linker within the motor domain that kind of flips the other foot forward. That is what is giving it the directionality. Here you have this neck linker attachment and everything, but there is no other foot that is moving forward. So for this motor, this single motor, it sometimes seems to backtrack, then it seems to be going forward. So ये जो sort of it seems more like a diffusion movement over a microtubule because ये directionality इसका इतना है नहीं. So now suppose you've taken multiple such events. बहुत सारी events इस तरह के आपने record किए हैं और हर एक event को आपने simplify कर लिया to indicate just the movement of this head. So suppose this movement, how do you track the movement? You track the movement with time, right? This is all snapshot with time. So if you have, say, on the x-axis, what did they do? Yeah, on the x-axis, suppose you track time. या फिर यहाँ पे for us to make it easy, let us say that on the y-axis we are tracking time because here these successive frames are 0.5 seconds apart. So you know that if you have on the y-axis you're tracking time and on the x-axis you're tracking distance, then you will have a line like this, right? If you just simplify this entire movement to something called a kymograph, which is with time you're looking at the position, you would have a simple line. Here you would have a line that once goes back, then it moves forward. So if you look at such successive tracks, then this is what you would get for the single-headed myosin motor, kinesin motor, and this is for the double-headed kinesin motor. Now, from this track, what is it that you can understand? Let me rephrase it once more. क्या किया है आपने? आपने ऐसे successive frames देखे हैं और आपने ऐसे सक्सेसिव फ्रेम्स में काइनेसिन मोटर हेड को ट्रैक किया है विथ टाइम. तो एक एक्सिस में आपका टाइम है और एक एक्सिस में डिस्टेंस है. So now you're able to see that this is the way the single-headed motor will walk. This is the way the double-headed motor will walk, right? And this is इस डायरेक्शन में टाइम है और इस डायरेक्शन में मूवमेंट है. तो अगर उसने मूव ज्यादा किया है तो वो ज्यादा ऊपर जा रहा है, अगर उसने कम मूव किया है तो वो ज्यादा फ्लैट होगा विथ टाइम. तो यहाँ से आपको क्या दिखता है? What is it that you can conclude from here? Try not to read what is written. Just try to interpret. Just try. There are no wrong answers. Just try. Anybody?
In the graph C, the movement seems unstable. As it is in such a zigzag pattern, and in the E, movement seems more stable than as compared to.
Okay, I could just get it partly. So you're saying one of them is more stable than the other?
Yes, ma'am. E is more stable in movement because the graph is more or less. It's not proper straight, but it is somewhat straight than the.
Right. So, right. Thank you. Thank you, Kavan. So the first thing that you notice here is that the single-headed motor is bound to microtubule for a considerably great time. So this is bound to microtubule. You see that if this axis is second, तो ये बहुत देर तक bound state में रहा है microtubule से. लेकिन यहाँ पे प्रॉब्लम क्या है? प्रॉब्लम ये है कि ये ज्यादा डिस्टेंस कवर नहीं किया इसने. Look at this, it rises very fast, right? These lines rise fast. और यहाँ पे क्या है? ये माइक्रोमीटर्स में है, इस एक्सिस में. So rising on this axis means it's moving fast and with time. In a short duration of time, your double-headed kinesin motor has moved a considerable distance. But single-headed motor is bound to microtubule, all right, but there is a problem in its motion. In the sense, it also sometimes backtracks. जैसे आप देखिए ये line ऊपर गया है, फिर ये नीचे आया है. That means it sort of sometimes takes a pause, goes back, then again moves forward, then again goes back, which means that इसकी directionality में भी problem है. So there are two things that you have understood here. from this simple experiment, it's a very simple experiment, just this setup is giving you two ideas. One is that the single-headed motors do bind to microtubule, can stay attached to microtubule for a considerably great amount of time. And that can also be seen here. ये पूरा जो भी उन्होंने रिकॉर्ड किया है डेटा, अभी उस डेटा को डिफरेंट वेज में एनालाइज किया है कि what are the number of motors that have stayed attached to microtubules, say for 2 seconds, more than 2 seconds, more than 5 seconds. इस तरह का उन्हें एक histogram plot मिला है. This is for single-headed motor. This is for a double-headed motor. So you can see both of them stay attached to microtubules. The single-headed motor seems to be attached to microtubules for considerably greater time, but it does not translocate. So if you look at the distance that it has moved, this color is for single-headed motor. The distance covered is much more by a double-headed motor. You can see it has moved a considerable distance, not the single-headed motor. Then they sort of hypothesized, and it's not just these people, but there are, as I said, there were a number of papers that came out in that period that hypothesized why exactly this, or how exactly the single-headed motor is working. So the first thing they said was that this motor head is positively charged, and it has a great affinity for microtubules which have negative charges, right? We do know that microtubules have a negative charge because microtubules के जो tails है, उनमें you will see a great number of these negatively charged amino acids. Microtubules are highly negatively charged. And the head domain of kinesins, being positively charged, have a great affinity for the microtubules. But the binding is kind of weak. And there is thermal fluctuation, there is Brownian motion. All of them give rise to the fact that although this it does bind to microtubule, quite stable, but because of thermal fluctuations, although it seeks the nearest next binding site, it can sometimes go back. It can sometimes go back because of these fluctuations that causes this molecule to sometimes backtrack, and then it finds the next say dimeric position in front, and then it walks in front. So this single-headed kinesin motor, इसका problem यही है कि इसका directionality नहीं है. ये नहीं है कि bind नहीं कर पा रहा या फिर ये microtubule के ऊपर stable नहीं है. Rather, it sort of has lost its sense of direction. So sometimes it backtracks, then it moves in front. Okay. Now, this was 1999, a very old paper that dealt with this question of a single myosin motor. Now, we come to this paper, which is, I think, 2019. And it's again asking the same question. कि एक single kinesin motor, how is it that that single kinesin motor is able to move across a microtubule? But then they ask a question, एक और question add कर रहे हैं उसके साथ कि how is it can now transport cargo across distances in a cell? Because what did we see initially? We found that the single-headed kinesin motor can bind to microtubule, but it has a problem in directionality. If that is the case, then why would nature produce single-headed kinesin motor if it is not good in finding its direction, if it is a slow motor, if it cannot walk well? Then why is it that nature has created single-headed kinesin motor? That means there is something about this kinesin motor that we do not understand. If it is there and if it is functioning, then there must be a way it has overcome this. So that was the question. कैसे ये long distances के लिए, how does it sustain movement of cargo? If it is a slow motor, if it backtracks every third or fourth step, then it is not going to be an efficient motor. So what is it that they did? So this again is the same thing as this. So what is it? It's another chymograph. What do you see? You see time on one axis, you see distance traveled on the other axis. And here you see a double-headed kinesin motor. Here you see a single-headed kinesin motor. Now, how did they produce these single-headed and double-headed kinesin motors? It's given here. But let's forget that. That's a construction of the motors, the way they did it. But if we forget all of that and if we just think that okay, this is the track created by a double-headed kinesin motor, this is by a single-headed kinesin motor. Again, what you see is that this double-headed kinesin motor on a microtubule is able to walk considerable distances. Very clearly it's moving and very fast. Whereas here it moves, but it seems that the movement is not that efficient. Because as we had seen the last time, they would move, they would backtrack, they would stall. So it's not very efficient in covering great distances. You see the same thing, it doesn't cover great distances. It doesn't rise on this axis. You don't see these straight lines, which indicates that ज़्यादा दूर तक ये cover नहीं कर रहा, लेकिन ये bound है. It is definitely bound to the microtubules. And what they see is that the movement here is more like a diffusion, because diffusion में क्या होता है? Diffusion में आपका direction तो होता है, लेकिन the diffusion is also kind of directed by Brownian motion. So it's the same thing that you see here. This kinesin motor doesn't have that directionality, which we have already seen. तो यहाँ पे हम लोगों ने एक स्टेप आगे जाके कुछ किया है, and that is what is interesting, and this is what with which I will conclude today's lecture. So let us let us look at this second experimental design. So the idea for them was to not look at things on a glass slide. Glass slide पे तो 99 में हो चुका है कि हमने माइक्रोट्यूब्यूल डाला, उसके ऊपर से काइनेसिन मोटर्स डाले, काइनेसिन मोटर्स सिंगल हेडेड बनाया या डबल हेडेड बनाया, दोनों को अलग फ्लोरोफोर्स से टैग किया, फिर हमने टर्फ माइक्रोस्कोपी के नीचे उसको देखा, तो हमने देखा कैसे वो मूव कर रहा है. तो ये तो हो चुका है. यहाँ पे इंटरेस्टिंग क्या है? यहाँ पे इंटरेस्टिंग ये है कि ये सेल के अंदर कैसे बिहेव करता है. तो सेल के अंदर अगर हम इस मोटर के साथ एक वेरी स्पेसिफिक कोई कार्गो को टैग कर दें और हम फॉलो करते रहे कि वो कार्गो कैसे बिहेव कर रहा है, तो हमें ये पता चल जाएगा कि सिंगल हेडेड वर्सेस डबल हेडेड काइनेसिन मोटर्स में कार्गो कैरी करने की क्षमता सेम है, नहीं है, क्या है. Question is, what happens to a cargo when it is tracked by a single-headed, when it is carried by a single-headed motor? So what you could do is you take a cargo and you somehow very specifically tag that cargo to this kinesin. And it has to be a very specific tagging so that you're not looking at the movement of this cargo by other kinesins, because there are other kinesins that could also be doing this. So how do you then design this experiment? And that's crucial here. So there are some mechanisms or some ways by which you can target a very specific, or you can induce sort of dimerization of specific domains. So there are multiple such methods in cell biology that is used. One of them is this FRB FKBP. So these are two domains from two different proteins. You take these two domains and you add something called rapamycin. Now rapamycin, ये पहले नाम सुना है किसी ने? Rapamycin.
Yes.
Yes, rapamycin would remind you of mTOR, right? So it's a target of rapamycin, TOR. So that's rapamycin. The same. Yes. So the same rapamycin, if you use that rapamycin, तो ये दो डोमेन है आपके पास. एक प्रोटीन के दो डोमेन है. ये वैसे इंटरैक्ट नहीं करते, लेकिन अगर आप rapamycin डालेंगे तो ये दोनों एक साथ इंटरैक्ट करेंगे. So very simple construct. You have two different domains. The two domains do not interact with each other. You add rapamycin, you're sort of creating a glue. The two domains will start to interact. अभी उन लोगों ने क्या किया? Peroxisomes है cells के अंदर. You have a specific target sequence that can target proteins to peroxisomes. So you take that target sequence so that your protein is now targeted to the membrane of the peroxisome, and you add one of this domain to that सीक्वेंस. तो क्या हुआ, पेरोक्सिसोम टारगेटिंग सीक्वेंस है, फिर ये एफकेबीपी है, ये दोनों एक साथ जाके पेरोक्सिसोम्स के ऊपर बाइंड कर चुके हैं. So it's present on the peroxisomes. Now you take your specific kinesin and you add the corresponding FRB domain to it. तो क्या होगा, जब रैपामाइसिन नहीं है तब ये डोमेन दो बाइंड नहीं करते, तो दोनों अलग रहेंगे. जब आप रैपामाइसिन डाल रहे हैं तब ये दो डोमेन्स इंटरैक्ट करना चाहेंगे. सो दिस डोमेन वुड देन गो बाइंड हियर, which means that your kinesin would be specifically linked to your peroxisomes. But you can control the timing of this event by adding or removing rapamycin. So very simple, beautiful way of doing this is that they took these two domains that do not themselves interact, but the moment you add rapamycin, they will start interacting. You add one of this domain to the membrane of a peroxisome, you add the other domain to the kinesin. Now your kinesin can be single-headed, can be double-headed. So your task is now to see कि rapamycin डालने के बाद पेरोक्सिसोम्स का पोजीशन क्या है, क्या वो एक ही जगह जैसे बैठे थे वैसे ही बैठे हैं या फिर दे स्टार्ट मूविंग अपार्ट. Is the experimental design clear?
Yes, ma'am.
Okay. All right. So what I'm going to show you now is a video from this paper, and what you're seeing is peroxisomes are these structures that you see here, these orange colored dots. And the moment they add rapamycin, if the in this the video that I'm going to show you is not for a double-headed kinesin motor, it's for a single-headed kinesin motor. They have taken a single-headed kinesin motor and they have added this domain to the single-headed kinesin motor. If they add rapamycin, then the single-headed kinesin motor would get bound to peroxisome. And if the single-headed kinesin motor is capable of carrying the cargo, then you should see movement of these dots. So let's see what they find. You see with rapamycin, all of them from the center, they end up going to the periphery, following the microtubule plus ends. Beautiful, right? Let's have a look at this video once more. So what does this indicate? What does this simply indicate? It indicates that this single-headed kinesin motor, हमने अब तक क्या देखा था? Single-headed kinesin motor का directionality नहीं है ना? In cell-free systems, जहाँ पे हमने microtubules डाला है एक slide के ऊपर, and we are looking at it, उसकी directionality नहीं है. इन लोगों ने भी वही check किया, कहा कि हाँ, इसके direction में problem है, काइनोग्राफ सही से नहीं आ रहा है, डिस्टेंस कवर नहीं हो रहा विद टाइम, बाउंड है माइक्रोट्यूब्यूल से, सब सही है, लेकिन डिस्टेंस कवर नहीं हो रहा. सेम चीज हमने डिफरेंट वे से सेल के अंदर देखने की कोशिश की. हम देख रहे हैं कि यार एक कार्गो तो मूव कर चुका है, सुंदर डायरेक्शन है, एकदम सेल के सेंटर से स्टार्ट हुआ है और एकदम सेल के पेरिफेरी तक पहुंच गया है, वेरी फास्ट मूवमेंट. तो यहाँ पे where is the what is it that we are missing? What we have missed is the fact that you are not looking at maybe one motor that is moving here, but multiple motors that are attached to the peroxisome. So the condition that we are looking at is multiple. It's almost like myosin. थोड़ा अगर मायोसिन के बारे में आप सोचें तो मायोसिन जब पॉलीमराइज कर जाता है तब आपका ये सेंट्रल मायोसिन का फिलामेंट है, बहुत सारे हेड्स वहां से नीचे आ रहे हैं. तो आप उसी तरह से इसको इमेजिन करने की कोशिश कीजिए. यू हैव अ पेरोक्सिसोम, यू हैव मल्टीपल काइनेसिन्स दैट आर बाउंड टू इट. All of them together is kind of giving it a process of movement to the front, not allowing the backtracking of those single kinesin heads that are bound to this cargo. In effect, ये processivity इसको सामने खींच के ले जा रहा है. सो तो आइडिया क्या है, आपका सिंगल हेड है, लेकिन हम सिंगल हेड का इफेक्ट नहीं देख रहे, हम देख रहे हैं बहुत सारे सिंगल हेड्स एक कार्गो के साथ बाइंड कर चुका है और वो बहुत सारे हेड्स एक साथ उस कार्गो को मूव कर रहे हैं। इन इफेक्ट इट्स सॉर्ट ऑफ giving it a directionality and helping in the very fast movement of the cargo from the center to the extremities. अभी उन लोगों ने एक और चीज किया, बिकॉज it's what should I say, you would have different kinesin motors with distinctly different lengths. So if we look at this, if you look at this, you would see that some kinesin motors are shorter when it comes to the domains other than the motor domain. Some of them are longer. So how does the kinesins, how do the kinesins behave in terms of load carrying capacity if you change these distances? So that was another question that they asked in this paper, which is an interesting question, and for which they had a very good way to see, because अल्टीमेटली वो क्या देख रहे हैं, अगर आप इस सिंगल हेडेड काइनेसिन मोटर के ऊपर एक ऐसा हेलिक्स लगा दें और इस हेलिक्स का आप लेंथ कंट्रोल कर सकते हैं जस्ट बाय कंट्रोलिंग द नंबर ऑफ रेसिड्यूज यू आर एडिंग टू दिस हेलिक्स, राइट? You remove the few residues, it becomes a few nanometers shorter. So if you can control the length, now you have the same system. आपको सिर्फ यही देखना है कि peroxisomes disperse हो रहे हैं नहीं हो रहे। तो वो क्या देख रहे हैं, if you just have the motor domain with the neck linker, you see the peroxisomes are beautifully dispersed. As you keep adding these helices to the end of the kinesin, and you keep increasing the distance between the cargo and the motor domain, the efficiency of dispersion of peroxisome or carrying away the peroxisome to the periphery reduces. So जितना आप ये distance बढ़ाते जाएंगे between cargo and the motor domain, you're reducing the efficiency of carrying it together. It's almost as if you think of it as a caterpillar. ये बहुत simple सा example दे रही हूँ। आप लोगों ने caterpillar देखे हैं? Caterpillar के क्या होता है? बहुत सारे छोटे 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 पैर होते हैं, हाँ? Very coordinated movement helps the caterpillar to move forward. Now, if you somehow you're able to increase the length, say tenfold, of the legs, how would the caterpillar move? Would the caterpillar be as efficient in moving forward, or would it be slowing down? From this experiment, you see that the caterpillar would be slowed down because ये जो whatever movement, it's dependent on the distance between the motor domain and the cargo, and if you increase this distance too much, which you can obviously ये हम imagine भी कर सकते हैं, अगर हमने बहुत ज़्यादा इस दोनों को distance बढ़ा दिया है, then इन दोनों में coupling में भी problem होगा। और ऐसा नहीं है कि हम एक मोटर डोमेन से इसको सामने ले जा रहे हैं, बहुत सारे मोटर डोमेन्स हैं, तो एक में एक के साथ दूसरे का कपलिंग का प्रॉब्लम होगा। The whole thing would be slowed down, and that is exactly what they see here. Okay. Now, last bit of information. So the same thing they tried to measure using an optical trap. अभी ऑप्टिकल ट्रैप तो हम 2-3 दिन से ऑप्टिकल ट्रैप के बारे में बता रहे हैं, तो ऑप्टिकल ट्रैप को अभी एक्सप्लेन नहीं करेंगे। What we're going to see simply is what they were trying to see. So the whole idea was to see that you have these single-headed kinesin motors, and they are able to efficiently tag along or efficiently pull a cargo. But they found that the efficiency was dependent on the distance between this motor head and the cargo. If you change this distance, then you have a problem with the efficiency with which peroxisomes are dispersed, and they are pushed towards the periphery. तो फिर यहाँ पे उन्होंने क्या किया, यहाँ पे एक बीड लिया है, सिंपल बीड लिया है, जिसके साथ ये सिंगल हेडेड काइनेसिन मोटर्स अटैच्ड है। Either this one, that is just the motor domain and the neck linker, or say something like this, which has a longer helical tail that increases the distance between the bead and the motor domain. फिर उन्होंने इस पूरे सेटअप को एक ऑप्टिकल ट्रैक के नीचे डाला है। तो क्या होता है जब भी ये काइनेसिन हेड्स इफ दे आर एबल टू एफिशिएंटली मूव the bead away from the trap, then they would move to greater distances and induce a greater force. And then the trap, because it's a trap, it would try to bring this bead back into the center position. So you would see some graphs like this. This is a graph for this condition. This is a graph for this condition. And you can very clearly understand that this is actually creating a much efficient movement of the bead compared to this condition, where you see the movement itself is not that uniform and is not that, what should I say, it doesn't work the same way. So yahan pe it starts building up, increases the distance that the bead moves on the microtubule, and then because it's a trap, it falls back into the trap, and you see it goes back in distance with time. Here you see there is an increase, not as impressive as here, and it falls back, and sometimes it rises and falls back instantly. So yahan pe, if you increase the distance between the bead and the motor domain, you are not creating the same force on the bead or on the cargo as you were doing with this condition. So with that, I will stop today's class. What we have learned today is that even if you have a single kinesin motor that is working on a microtubule, it is still, as you see here, it is still highly processor. With time, it still stays on the microtubule, doesn't fall off. But it has a problem in directionality. It tries to backtrack. So it doesn't fall off, but it goes in front, then it backtracks. Then it goes in front, it sometimes backtracks. So the backtracking is very prominent. It goes in front, but then it sort of backtracks. All of this can be overcome if you think of a situation where you have a cargo that is bound to multiple such single-headed myosin motors. In that case, it becomes a very efficient system in moving that cargo towards the, say, the plus end of the microtubule. And this is a very highly efficient process of movement, and this is dependent on the distance between the cargo and the motor domain. If you increase this beyond a certain limit, you'll see that the movement is there, but the force generated on the cargo is not optimal. Okay, with that, let me stop today's class. And if you have any questions, I'll be happy to answer. Maybe I've overshot the class. I'm not sure how long. Wow, 6 minutes more. Okay, if you have any questions.
Yeah. How do group of kinesin motor proteins move in a group?
How do?
How do this group of the monomers kinesins move? As in, do they slide or or they go by hand in hand, like you explained for diamers?
They do not slide. Sliding movement nahi hai. You have to remember that there is a definite spacing on the microtubules where they can bind. So every time they move, they have to find the next binding region where they can bind. They will not bind arbitrarily, right? तो आप इस तरह से सोच लीजिए, रेगुलर लैटिस है और रेगुलर लैटिस में रेगुलर स्पेसेस में इनके पास जगह है जहां पे ये बाइंड कर सकते हैं. And if you think of a single head, then the single head is going to try to find its next binding site, and that's the reason it can sometimes backtrack, क्योंकि next binding region इसके पीछे भी हो सकता है. So it can sometimes backtrack. But if you now have multiple such heads on a cargo, तो तब ये really एक directional movement बन जाता है, where it keeps looking for the next binding site in front, because you have multiple heads that are now tagging the cargo along on the microtubule. So it's not a sliding motion, it is definitely searching out for its next binding site. every time.
Okay.
So step size would remain the same. Step size में difference नहीं है. 8 nanometers, 8 nanometer. Okay. All right. This was kind of a food for thought for all of you, so that you have some idea कि kinesins कैसे हैं. This is an extreme example of where we are looking at a single-headed kinesin, not a double-headed kinesin. So think over it. Look at the slides. I will upload it today on Imparter's backpack, and tomorrow we'll go a little bit into dining. And Friday class नहीं है क्योंकि Friday मुझे gate duty पे जाना है. So tomorrow is going to be the last class, and whatever I teach tomorrow, up to that, it's going to be your syllabus for your minor exams. If you have any questions, anything regarding the motors, please do not hesitate to send me a mail. I will reply as soon as I can. All right. Thank you. Thank you so much.
Thank you, ma'am.
Thank you.